Hi.
Hi.
Woo. I'm. Oh my God. Okay, I knew I'd get that wrong. I'm terrified of those echoes. Hi, I'm Valda Vinson, and I'm executive editor for the Science Journals.
And I'm Alex John London. I'm the K&L Gates Professor of Ethics and Computational Technologies at Carnegie Mellon University.
Okay, thanks. So on behalf of the full planning committee, who is sitting around here, we want to welcome you to this workshop on navigating the benefits and risks of publishing studies of in silico modeling and computational approaches of biological agents and organisms. Quite a mouthful. So we want to thank the planning committee here, the National Academy staff, and today's speakers, all of whom have worked hard to bring together this very exciting event. And before we dive into today's goals and objectives, Audrey Tevenon from the National Academies will give us some brief opening remarks and also a little housekeeping information. So Audrey, the floor is yours.
There's a lot of logistics involved in this meeting, including turning on my mic. We are using a regular Zoom. This is not a webinar that you sometimes have an opportunity to see. You are muted. If you're online, you are muted on entrance. In the breakout room, you will have the ability to have the mic and access if you have checked of being an active participant. People in the room, of course, if people in here will have the opportunity to also have the mic. If you're on Zoom, please update your last name and first name by clicking the more. We will use Slido in the room. If you have a phone, there is a Slido QR code that is around. We can use this. It's also on the slide here. We will be gathering the questions through that platform. This also is a platform where you get to vote if you find that a question is particularly of interest. In the breakout rooms, we'll use Padlet. This is another way of collecting some input. Everybody in here or online will have a chance to contribute to this, this activity. If you need assistance, contact Deja that is on this slide. The workshop right now in plenary session is recorded. However, you will not be recorded during the breakout session. The speaker biographies and agenda can be found also on our website. We have captions for people online. This is how you will do it on the slide. This is an hybrid event, so it comes with a lot. We ask that people just show some respect for each other. It is easy when you're in the room to be talking over people who are online. I can emphasize more that we want everybody to have the best experience. So it is here, and it is especially in the breakout room. We want you to stay open to each other's comments and include all the voices that are around the table. We're trying to assemble a very diverse group of stakeholders, and we want that to be reflected also in the way we talk to each other. Use the raise button if you're online during the breakout to talk, and then here in the room, you can raise your hand and you have a mic, or you have the podium also has a mic. So that's all I have. And after this, I'm gonna turn it to Balda. Thank you.
Thanks, Audrey. So now on to the goals of the workshop. So today on day one, the sessions will be focused on discussing how existing policies and guidance and safeguards that were mainly developed to address experimental life sciences research with dual use concerns also applies to in silico studies, particularly in the context of disseminating those studies. Are there challenges and gaps around responsibly disseminating computational studies, models, and tools? And so notice our focus here is really on the dissemination of the studies. So, and today really looking at what exists and what are the challenges and gaps. And then tomorrow on day two, we'll build on day one, and we'll hear some case studies that have had to consider biosecurity challenges in the context of disseminating the studies, and then discuss specific best practices that would safeguard the benefits of the research while reducing the risks of disseminating that research. The 2 days are really aimed to be integrated and to build on one another. So please plan on joining us back for tomorrow, because that's really when all the outcomes will be discussed. Okay, so the workshop is designed to be highly interactive, and it will include case studies and group discussions reflecting researchers, publishers, government stakeholders, and many others. So all of these stakeholders have to work together to move the conversation to a place where we can promote science and innovation while ensuring that we have safeguards in place. We expect that the workshop will pave the way for suggesting strategies that can be implemented by the relevant stakeholders to reduce the risks and enhance the benefits. So thank you. And I think we get started with Alex, who will present the charge for the planning committee and will really frame the workshop more specifically. Thanks, Alex. The floor is yours.
Wonderful. Thank you so much, Valda. Thank you, Audrey. I have a few slides to set the stage for the event today. In 2024, the United States government issued new policy guidance for the oversight of dual-use research of concern, DURC, and pathogens with enhanced pandemic potential, PEP. That document is focused on the oversight of physical, laboratory-based research, but near the end, it mentions the importance of developing voluntary guidance for activities that are out of scope for that report. These activities include in silico models and computational approaches in biology, and the importance of developing guidance to assess the benefits and risks of such research, and for the responsible sharing and communication of research results and data sets. The importance of developing voluntary guidance for in silico models and computational approaches in biology is reiterated in the implementation guidance for this policy, where there is once again a focus on assessing benefits and risks of in silico research, and the need to develop practices for determining responsible communication of results, models, and data sets associated with such work. The statement of task to the national academies was thus to convene a planning committee to facilitate this workshop, where our charge is to consider the benefits and biosecurity risks of communicating studies involving computational modeling and the analysis of biological systems. Given the diversity of research outputs for in silico research and the venues or outlets in which these outputs are disseminated, we have focused on the broader category of dissemination rather than publication. Our task for the next 2 days is thus to consider existing guidance that is relevant to the communication and dissemination of in silico research, the challenges and needs related to safeguarding the benefits of this research while mitigating its associated risks, taking into account the evolving efforts on AI safety and communication in order to identify policy options and norms for safeguarding the benefits and advances of this rapidly developing area of science while reducing relevant risks. So to this end, we've assembled a diverse group of experts from a wide range of disciplines, from the academy, industry, government, and non-governmental entities to facilitate this task. So a philosopher can't go anywhere without a Venn diagram. So this is a very large task. As a result, it's crucial that we understand how the workshop is situated within a larger web of issues so that we can focus on questions that are in scope for the meeting. So in that regard, this workshop takes place at the intersection of these 3 broad areas. It's focused on in silico models and computational approaches to biology. It's focused on how to preserve the benefits of this research while managing associated biosecurity risks. And it's also focused on both of those things when it comes to the dissemination of research outputs and resources. So to further refine this focus, in this slide, we've tried to capture the questions that the workshop will seek to address across the 2 days. The polygons on the diagram represent specific questions that we hope to clarify. So with my last few minutes, I'm going to walk through this in a bit more detail. So just to be clear, a reminder about 2 points. The first is that this workshop doesn't focus on upstream oversight mechanisms that ought to govern the conduct of in silico research in biology. Those mechanisms are out of scope for us, as our focus is on benefits and risks associated with the dissemination of research. The second point is simply that some of these upstream mechanisms are relevant to the conversation we'll have because they either constitute a form of dissemination or because they impact some aspect of dissemination. And one unique aspect of in silico research is that it has a wide range of outputs and associated resources, which can be disseminated through a wide range of outlets, much wider than the more traditional focus of publications and journals as an outlet. So one of the goals of this workshop is to make sure that we can identify which outputs from in silico research are relevant to our task, then which outlets for dissemination are also relevant to this task, and then who the associated stakeholders are who have responsibility for safeguarding the benefits of such research while mitigating and managing the associated risks. So just as a way of priming the pump, one set of questions for day 1 involves capturing the relevant research outputs. So this might include peer-reviewed papers and preprints, but it also includes working code or final code or models and data and user interfaces. And then our question is, what else? What are the other outputs? Outlets for dissemination can include journals and preprint servers, data repositories, working environments, applications with which users might interact through an API. What are the other outlets that are relevant? And then finally, the stakeholders might include authors, editors, reviewers, industry, researchers, data and code curators, funders, users, who else? So this is one set of questions on the agenda for day 1. Another goal for day 1 is to discuss the criteria that should be used to assess and evaluate research benefits and associated biosecurity risks. So on the one hand, we don't want to reinvent the wheel. There are existing policies and guidelines that are relevant to our task. However, existing policies in this space tend to focus on physical research rather than in silico research. And they tend to focus on risks associated with dual-use research of concern, DURC, or pathogens with enhanced pandemic potential, PEP, often as these risks arise from the conduct of that research. As a result, one of the goals of the breakout session on day 1 is to discuss current challenges and gaps in assessing and evaluating the possible benefits of in silico research in biology, and the DIRC and PEP potential from the dissemination of that research or its associated resources. Do different outlets for dissemination pose different risks? Are there additional criteria, if any, that should be used to assess the biosecurity risks of in silico research in biological systems for each of these outlet and output types? Finally, the focus of day 2 will be to consider lessons learned from other technologies in other research domains across different sectors, and then consider what approaches or strategies should be used to promote high quality science, scientific progress, and the openness of science while mitigating biosecurity risks associated with disseminating in silico research in biological systems. So I want to emphasize that even if there is disagreement about the issues that we've talked about so far, these disagreements likely take place against the current status quo in terms of in silico capabilities, and these might change in the future. So it's worth considering what different strategies might be used by various stakeholders to mitigate biosecurity risks, even if we disagree about the nature and magnitude of those risks. I also want to emphasize that these strategies need to protect the integrity of science, the openness of science, and promote the benefits of scientific progress. So identifying strategies that balance these goals is a valuable contribution. So this is meant to be an interactive workshop, and each of the boxes on this diagram is a place where we are seeking your input. So this isn't meant to foreclose the conversation, it's meant to focus the conversation. So here's the agenda at a glance with the 4 sessions that we have planned to help us advance the goals for the workshop on the first day. So now let's turn to the first session of the meeting. The goals of session 1 are to set the stage broadly for our current state of knowledge regarding biosecurity, including DIRC and PEP policies, advances and innovations in research using computational approaches, and their applications in biological systems, all of these in the context of publication and dissemination of in silico research and resources. So we have a fantastic lineup for this session with 5 speakers. In the interest of time, all of the biographies can be found on the website. And so that link to that will be put into the chat. Each speaker for this session will have 10 to 12 minutes maximum to provide their thoughts, and we will open the floor then to question and answers. Audience members are invited to submit their questions anytime using Slido, where everyone can then upvote questions they most want to hear answered. So the Slido link will also be put into the chat, and we'll address as many of these as possible in our discussion. So it's my pleasure to turn now to our first speaker, Gigi Granval, who is professor in the Department of Environmental Health and Engineering at Johns Hopkins Bloomberg School of Public Health, a senior scholar at the Johns Hopkins Center for Health Security. Gigi will provide a high-level overview of dual-use research of concern and pathogens of pandemic potential guidance in the context of publication and dissemination. Professor.
Right. Well, thank you so much for the introduction, and thank you so much for inviting me to speak, and thank you to the staff to handle all the questions that I had coming here. So I was asked to give a little bit of context to the concerns but that information could be misused. This is not a new problem. So, and it goes much longer ago than I'm going to start, which is during the Cold War, when people who were studying physics, publishing in physics, some classified work in physics was demonstrated pretty clearly to be aiding the Soviet Union in their in developing missiles, and so it was actually seen as the major part of their program. So that was obviously a problem. And the National Academies formed a committee chaired by, it's now known as the Corson Report after the chair, where they studied this issue to try and look at the benefits versus risks of information being out in the world, scientific information being out in the world. And they came to a conclusion that security by achievement was much better than security by secrecy, and that it was really important for the scientific process to have something to demonstrate achievement, to have that openness, and to and even in the case of nuclear weapons development. Fast forward some time after 9/11, the National Academies again played a really big role, funded by the Sloan Foundation, where they, after 9/11 and the anthrax letter attacks, talked about how research could be misused, that research that's done for legitimate purposes, but nonetheless could lower the barriers to biological weapons development. And this was the report that came up with the 7 experiments of concern. So things that you might do for legitimate purposes, whether it's for vaccine development or animal model creation, that nonetheless could lower the barriers to biological weapons development. And around that time too, journal editors came together to talk about how they would handle the publication dissemination of research that was dual use research of concern, one of the many acronyms that we have in this space. So the problem, however, is that we don't have a biological weapons development program. We have no offensive program where that information is where people would actually be trying to do that. So all of these things have legitimate purposes, the scientists that are publishing in this space, that could be misused. So they have this flip side, and so the information needs to get out there to advance science. Fast forward another decade or so, and there were 2 virologists or 2 virology labs, Fouchier and Kawioka lab, that tried to publish their work on what became known as gain of function pandemic influenza. In retrospect, those mutations that they identified were very important ones for our understanding for how H5N1 could become, at that time, transmissible in mammals. Now we know, of course, that it is transmissible in mammals, and we are worrying that it could spill over further into people. So, but the concerns of the National Science Advisory Board for Biosecurity, or NSAB, at that time, an HHS advisory body, that their concerns were that this work could be misused, that it could be used to develop a biological weapon. Their concerns were entirely security-based initially. And they sought a way to share this important public health information that is important for surveillance of H5N1 with people who needed to know, and to somehow exclude the people who didn't need to know. Problem is, that's really hard to do when this is an international problem, and our classification system exists to, they're more interested in your travel history than your expertise. So this is a challenge that has been continuing to this day. I think the current example and challenges with H5N1 in cows and dairy cows and migratory birds demonstrate that there is a lot we do not understand about pandemic influenza. And I believe that the fact that we do not know more about this pandemic threat is tied to the fact that it's been so hard to do research in this area. And so there is a cost to being able to put barriers towards a legitimate scientific inquiry. I want to say that right now we are faced with a similar kind of issue, and that's why this workshop is being held. But I want to say that I think that all of these things demonstrate the limits of prevention, that there has to be some acknowledge that the problem that of biological weapons, whether they are in the hands of the nation state or a non-state actor, is not something that can be solved only at the front end. You have to also have preparedness and response capabilities, because my second point is that there's a lot of people there's a lot of intelligent, clever people all over the world. And some of those people also want to do harm. And so preventing them from accessing this information is going to be always a challenge. And creativity is not something that's only found here. I'm not sure if stopping research on the prevention side has anything to do with why we are currently witnessing the complete dismantling of US technical and scientific progress at this moment. I don't think that is the motivation, but what we will see is that if work is not done here in the US, it will be done elsewhere. And we will have, we will be wondering we will be trying to get information that can help us in our scientific program, whatever it is at that point. So I think that the challenge is, how we can not help our adversaries will have limitations, especially if we are not in the game of actually producing research. I'll end there and take questions later.
Okay, thank you very much, Gigi, for those excellent comments. Our next speaker for this session is Linda Stewart, who is President and CEO of the Fund for Science and Technology. Today, Linda will talk about advances in computational models, foundational models, generative AI for complex biological systems, and available dissemination tools for the research results and resources.
I'm going to try and share my slides. Oops, probably sharing the wrong thing. Yeah, probably seeing my calendar. Don't want to see that. Does that look right? Yeah. So I wanted to just talk about two things or cover two kind of broad topics. So we'll try to get into presentation mode. Which is first, just to give you a very brief overview of a report that we just put out. Gigi is actually one of the co-authors, and Mike Imperiale, my co-chair from the NASEM study on AI in the life sciences, that speaks a little bit to the biosecurity benefits and risks, and then also just dive a little bit deeper into the technical on biodesign tools, because I think it's important to kind of help frame a little bit of this question. So this is the report. I would just encourage everybody to just maybe try and read it. But this was a DoD-sponsored report on the biosecurity, the benefits and considerations in the of AI in the life sciences. It was really triggered by the convergence of the advances in artificial intelligence and synthetic biology and life sciences. And really this recognition that the computer and information sciences have been evolving really very rapidly over the past decades, but we've seen this sort of convergence with artificial intelligence and AI-enabled biodesign tools for biological applications that had kind of come together in a sort of synergistic and amplifying way. These, as Gigi was expressing, these tools really have a lot of promising applications for drug discovery, medicines, synthetic biology for biologics, and other for drugs, for environmental mediation. We could go on and on and on. So there's a lot of benefits, but there are security concerns. So this is considered a dual-use research technology, where these tools could potentially be used to engineer biological agents that might be a threat, and that their wide availability might lower barriers to entry, the expertise or resources required. I think the power of the tools have been recognized last year in the Nobel Prize, and I'll talk a little bit about the work of my ex-colleague David Baker in chemistry for protein design in the second half of the talk. I think a really critical thing to understand as we are trying to have this discussion about the in silico question is exactly how are these tools used? So, just as a very, very quick primer for synthetic biology has these two phases. There's a sort of ideation phase and design phase where you're trying to think of a solution to something. And then that designed molecule has to be translated into the physical world through genetic expression of that molecule or that agent. And this is like just the design, build, test, learn principle that really underpins most of synthetic biology research. And what we did in the report was to actually ask the question, where does AI impact this end-to-end process? And what we really, when you look at it, what you'll find is that AI is at this point in time, is most impactful in the upstream. So in the ideation phase, where you have a large language models that can crawl a large amount of data, for instance, can give you many more ideas on how you might do something, or including beneficial and harmful things. And that the AI-enabled molecular design tools are really very powerful in designing proteins. They're not yet there for designing replicative agents, but they're very, very powerful in that realm. But those designs are really just generating a hypotheses that are on a computer. They are not actually in of themselves dangerous. And only when they are transferred into the physical world would you really have to worry about them as being biological agents. And that AI has not yet shown a massive uplift in our capability to make more things that may come with AI and automated labs, but we're not really there yet. So the report recommended that AI application is a very, very dynamic area. Things are moving very quickly. The capabilities for what you can do is limited by how complex the task is. So it's, they're very good currently for simple tasks like designing a molecule. They're less good for really complex experimental tasks, such as making a replicative RNA virus, as an example. And they still require experimental validation. There's high quality biological data is required for training those models, but there's limited data for training certain things, such as how to make a highly transmissible virus. We just don't have that sort of data. The beneficial applications are very significant. It's for medical countermeasure development, as an example, but that they are risks, but that the current capability uplifts primarily sit in the designs phase and not in that later phase. And that we must be really aware of the importance of these tools and these approaches for actually being mitigation strategies. So vaccines and medical countermeasures are accelerating the response to that. So that's the report summary, but I want to just talk a little bit more detail about the biodesign tools. And what I'm going to show you is really some work from David Baker's lab at the University of Washington, where the Institute of Protein Design, where I used to work up until about 4 weeks ago. And so just for those who are not familiar with this, there are two major kind of challenges that have been facing people in the protein structure world. First was, is it possible to predict structure from genetic sequence? And that's shown on the left. So can you take a DNA sequence and know how it's going to fold? And then the second was the sort of reverse problem of, can you design new proteins? And the reason for doing that would be to allow us to access totally novel protein space and not rely on proteins that existed in nature. So the left was the left protein structure prediction problem was solved by AlphaFold and soon after by RosettaFold in 2021. And then David's work on over a number of years, but accelerated by artificial intelligence on RF diffusion in 2023, really cracked the protein design problem. And how those models work is shown here, is RF diffusion. I'll show you the details of this in a minute. But essentially, it's a generative diffusion-based molecule, which will create a new shape. And then once you know what that shape is, you can assign different amino acids that you predict are going to fold into the correct structure. Those are only predictions, and you get multiple shapes through diffusion and different options through protein MPNN. And what you have to do is filter those to know to choose which of those you want to actually do experimentation on. So all of this happens in silico, but there is this phase after you've filtered with Rosetta Fold to actually then kind of go into the wet lab that is needed, where you go into the wet lab to understand whether you've created the right thing. So the thing about RF diffusion is a generative model. It's a bit like DALI or Midjourney. And what these are neural networks that have been trained on vast amounts of data. And then those neural networks have undergone a process of diffusion where it's really easy to explain. In the upper panel, if you take every picture of a cat and you develop a neural network that understands that information, you can then inject Gaussian noise so that the picture of the cat becomes increasingly pixelated until you just have pixels. And then you can take, if you run that network in reverse, you can take pixels and you can say, from these pixels, generate a picture of a cat, and that trajectory will give you a cat. You can try and prompt that trajectory by saying, give me a picture of a cat that's lying on the floor, or is purple cat, or whatever, right? So we've all used these tools. They're very useful. But every time you run the trajectory, you get a different solution. So you get vast amounts of theoretical solutions from a problem. So the same thing is exactly true for diffusion-based protein design tools. So you take an amino acid cloud showed here on the left, you say, design me a protein, and it will design you a protein. And every time it runs a trajectory, you'll get a different design. And so you get a vast amount of data that you then need to filter through using some sort of filtering model. So if it's to select which of the designs you want to go off are valid and behave as you think they should. So that filtering step is extremely important in down selecting from all this noise, all these hypotheses that you generate, to ones that are actually worth experimentally testing. So these are the sorts of things you can do. So these are just some designs from the Institute, peptide designs, mini binders, antibodies, and vaccine designs. And really importantly, these are, I have somewhat deliberately selected things where you could see how these designs may be used to develop medical countermeasures, because it's actually really important to appreciate that it really is almost out of the ether that you can now generate something that could block an antibody, for instance, that could block and bind to the influenza hemagglutinin, or a new vaccine. And that power really speaks to how much this benefits countermeasure development. So just going back to this picture, I think, and just to in the last couple of slides, just to frame this discussion for today, what is really critical is that the AI tools that we are talking about are generating hypotheses. They're not generating things in of themselves. Those things must be synthesized. And the question being asked is, should you publish or not those hypotheses? So just to summarize, I think synthetic biology and biodesign really is an extremely promising area of research. The upside is extraordinary. The US is currently a leader in the field, and I hope will maintain its global dominance in that, but who knows? The opportunities for impact are really quite enormous, and particularly in the wake of the new tools. Currently, I would say the benefits outweigh the risks, and innovation should be supported. In the NASEM report, we discussed managing the risk by adopting an if-then framing to monitor new advances. For the in silico question, I think the problem or we need to tackle is AI tools can generate vast number of hypotheses, but the risk for synthetic biology does not manifest in the design phase, but only when you move from the digital to the physical world. So currently, wet lab experimentation is required to validate designs and confirm function. It is possible to imagine at some point in the future that both design and filtering reaches a level of accuracy that wet lab testing is not required, but currently that is not the case. And maybe one thing I might propose to consider is for publication is to adopt a bit of an if-then solution for evaluating how and what to publish based on one, what is the proposed function that is being designed for, and two, by continuously evaluating the state of accuracy of design models and tools to understand whether wet lab testing is actually needed to validate those tools, or whether you're spitting out 100% of your designs are actually going to do what you think they are going to do. But we're not there yet. Anyway, so I'll stop there and take any questions.
Great, thank you very much, Linda. Those were extremely important insights. Our next speaker for this session is Sarah Del Valle, who is the Chief Scientist at Los Alamos National Laboratory. Sarah will talk about in silico research for biological applications with dual use potential and the way that research results and resources are shared.
Hello. Okay, let me try to share my screen. Share. Can you see my slides?
Yes, we can. Thank you very much.
Okay, great. Good morning, and thank you for the invitation to be here. I'm Sarah Del Valle. I'm a chief scientist here at Los Alamos, and I'm also a computational epidemiologist. So, oh, I don't know. Anyway, I wanted to talk about how we can navigate the dissemination of computational models. Okay, so I wanted to start with this statement, which I think, it's probably preaching to the choir, but we all understand that we're at a time where powerful biological designs and threats may not come from nature, but from code. And whether it's predicting outbreaks, which is one of, I'm sorry, it seems like I have some timer. Whether it's predicting outbreaks or generating entire genomes, there's a shift towards biological and in silico biological research. And so we need to understand the risk. So I wanted to start with a case study, which is my area of research, agent-based models, and I wanted to kind of show the risk and the benefits of using agent-based simulations. And since agent-based simulations have the roots here at Los Alamos, I felt like this was a good case study to demonstrate. So I want to start with von Neumann and Olam, that they came up with this concept of cellular automata, and they were thinking, von Neumann was thinking about self-replicating machines. And I think what was really interesting is that very simple rule-based entities interacting on just a grid, on a two-dimensional grid, can come up and produce very complex global behaviors. And so that emergence of complexity, I think, was very interesting. And that gave the foundation to many different other ideas, such as Conway's game of life, and which was able to model complex emergent behavior from simple agents. And so now, fast forward, we now have agent-based models. I'm just going to go all the way kind of here to the end. Agent-based models that are computational simulations where individual or agents, whether it's people or pathogens or vehicles or particles, they interact in realistic environments. And they're obviously guided by rules and decision-making processes, or although nowadays with the use of AI, they can also adapt and learn. And unlike comparamental models, which are system of differential equations that kind of capture a system at an aggregate level, these ABMs can model very complex emergent behavior from simple agents. And so now here at Los Alamos, we have kind of taken a lot of these computational resources and expertise to build national level simulations. One of them is EPICAS, and where we incorporate detailed demographics and mixing environments like school, workplaces, behavior, and these are all matched to the real world. And so these tools are used for both preparedness and response. We have helped with the COVID-19 pandemic and many others, but they can also have the potential for misuse. And so here, let me show you a video of some of our simulations that we were running during the pandemic. And so here you can see how the spread of the virus was simulated starting in April. And we were asked to consider the possibility of lifting the policies on June 1st. And so you can see not just the spatial scale, but also the temporal kind of scale of the model. And so I think that the take-home message is that these models are not just theoretical tools, but they can generate real-world biological implications, and they can identify transmission paths. They can help simulate intervention failures. They can also characterize the dynamic and the distribution of outcomes. And so they have this dual kind of use, where they're currently used for public health purposes, but they could also be potentially used. They can potentially be misused and be a vulnerability. And there was already a presentation on the challenges, but they're too narrow, and then they don't really capture some of the implications of these computational models. And so we need to figure out whether the requirement of journals now to release models and code. I think it presents a challenge for models like this computational model that I'm showing. So when I talked to the organizers about this presentation, they were thinking about kind of thresholds that I see in how we could use these models and what we could do to understand the risk. And so I put together kind of like a few questions that maybe can help us, that will help during the discussion session. But the one is on capability-based risk. Does the model enable enhancements? And specifically, because for my work is on algorithms and simulations that can optimize and automate a lot of the biological and social realism. The other one was exploitation risk. And so identifying vulnerabilities in human, animal, plant, ecological systems or infrastructure systems. In our case, with agent-based models like EPICAS, we can do this, and also training or fine-tuning models for malicious purposes, such as optimal transmission pathways. And another kind of threshold that I wanted to consider was also the intent and misuse enablers. And for this particular kind of models, like model societal impacts of specific attacks and optimal timing and targets. And so in coming up with these kind of questions, I was thinking about understanding the progression of risk dynamics. And so in the age of generative AI and figuring out global security impacts, we can start with the low complexity kind of linear risk. And so, think of malware, for example, which, they're bad, but their impact is more linear. And then you start going in the earth progressing to exponential, and think of a wildfire that is very hard to contain because of its magnitude. Then iterative, which is optimization and refinement of a specific goal. And then finally, the wild card, which I think this one, it's a little bit, maybe AI, it could be maybe in this category of discontinuous where you could be, you know that it could be in many different kind of places. And so because of I'm here at Los Alamos, I also thought about presenting a success story. And I know Professor Growell, Gigi presented at the beginning kind of the connection to the war in the 1940s. And I know that we have a global framework for regulating nuclear activities. And so there's kind of the different pieces of the regulatory body, which starts from prevention of proliferation, monitoring, verification, global risk, controlling nuclear trade, and banning nuclear testing. And so each of these pieces kind of play a different role, you know, from setting norms, such as the NPT, to verification and safeguards, the IAEA, and the Global Threat Reduction Initiative too, and the NSG, the Nuclear Supplier Group, that also control the trade of sensitive materials. But I think what's important about this is, and while it may not be perfect, is that it's structured, is resourced, and is international in scope. And I think as we continue to move forward to figuring out how we disseminate code and research, I think we do need these kind of three ingredients, like having it structured, having resources and funding, and also the international component. And then finally, the tools to simulate biology are outpacing the norms for sharing it. So it's time to rethink how we assess risk in the computational area, but also thinking about our responsibility also as researchers to disseminate our work. And so I think that we need to preserve openness without enabling risk, and that's what we need in a new framework is openness but without enabling risk. And we can calibrate this for computational models and ground them in lessons from other domains, such as the nuclear science. And I think that's all I have. Okay, let me stop sharing.
Great, thank you so much, Sarah. That was excellent. So the final set of presentations in this section will be Val DeVinson and Philly McGowan from our committee. They'll tag team to provide a publisher's perspective on the dissemination of biological research involving in silico models and resources, and where questions remain.
Excuse me. Thanks, Alex. So I'm Phelan McGowan. I'm an associate professor of biomedical engineering at Johns Hopkins University. I run a computational lab, but I'm also co-editor-in-chief of PLOS Computational Biology. Our journal is actually turning 20 years old this year. It's founded on, guided by the principles of open science. And as the workshop title suggests, we're looking at both the benefits and the risks of dissemination. So I'll start with talking about benefits and the reason we want to share more and more, and then Valdo will discuss considerations about the risks. So there's multiple reasons why open sharing of code and other research tools is important. So for example, accessibility is really important, right? So open science dissemination makes material available, accessible. Ideally, this accessibility should be author independent, so it doesn't depend on someone on request searching for a digital file needle in a haystack from years ago, but instead is available in accessible repositories with a persistent identifier. It's important that it be transparent, right? So that's one of the benefits as well, is being transparent. The evaluation of work by reviewers and editors and readers benefits from the open dissemination of the underlying code and other materials. And it helps them to better understand the details and check for errors or faulty assumptions. Reproducibility, another benefit, right? So going beyond reading about it, actually sharing the code and other materials facilitates reviewers, editors, and readers to directly recreate the work without excessive effort and see that the claims in the paper are supported. Then reuse, right? So scientific progress, you go on the next one. Scientific progress really, depends on this sharing, right? We build on each other's work. Progressive science often builds on the work of others, and open sharing and dissemination really increases the ability for other scientists to use, to extend, to enhance, build on the materials, and advance science even further. This inspires new directions, right? Prevents reinventing the wheel, even helps continuity of projects within a lab. And also, another benefit is that this is an equitable, right? This is equitable. Open sharing increases access to tools for scientific progress and makes that accessibility broadly available to all, rather than it being held closely or gated for a few. Now, a key point I want to highlight here is that open science, and particularly sharing and dissemination of codes and methods, is by and large a central part of the computational community's culture. And I think there's higher rates of adoption of open science principles in computational fields than in many other fields. When our journal introduced a new policy in 2021 that required authors to share code, some were actually concerned that this new requirement would decrease submissions, right? That it would create a barrier. And instead, the computational community responded really well, I think because it matched well with their own principles. So the policy empowered editors and reviewers to ask for code, and the journal went from about half of papers sharing code to over 98% sharing code by the end of 2023, so just within a couple of years. Submissions overall went up as well, and they've continued to climb. So sharing, dissemination, definitely central to computational research culture. So these are some of the benefits, the reasons, the imperatives for sharing and dissemination. And I'll just leave you with this. I said right there that we got to over 98% of papers sharing code, but not 100%. And the reason it's not 100% is that there are risks and other considerations. So I'll pass this on to Violet to talk about those.
Thanks. So I'm going to I think we heard in the previous two talks some of the risks around sharing code, and I'm going to start off sort of more general about when we allow exceptions to public access, basically to data and to code and even to materials. So I think hopefully much of the publishing industry has moved towards more open science. PLOS has been a leader in that area, and we've certainly done a lot to be more open, to move to being open. But there are these real needs for exceptions. And so what do we get people coming to us and saying, well, we can't share our data, or we can't share our code, or we can't share the weights for our code because, the one thing that comes up often is basically IP, and that's just a hard no from us. You cannot not share to protect IP, but we do. There are protections that come from patents and licenses. So work can, but with data, that's a bit tricky. To the best of my knowledge, there's not really sophisticated licensing around data. So our requirement remains that data has to be available, and as Phelim said, it needs to be available at a publicly accessible site. So that's what we do. But then so IP, no, not an allowable exception. What do we allow? The one that comes up with us most often that we do allow is privacy. So, and this is best known in clinical data. And there, I think this is where, clinical data, there are some real strategies in place that are used by everyone. And so we know if we get a paper in with clinical data, this is what needs to have happened. It needs to have been pre-registered at blanking, but yeah, clinicaltrials.org. Sorry. So it needs to have been pre-registered. The data, the data needs to be available in aggregate, and there needs to be a way to access at least the anonymized data. But that access could be through restricted access. And there are sites that are specifically set up to allow restricted access to clinical data. By researchers who can show that they have a legitimate, are doing legitimate research and need that data. So that's is a case of where, you know, there's a structure in place. And so when the paper comes in, we know what we have to do. Ecological data, you know, there is some data like geolocation of endangered species that you really wouldn't want putting out there. That mainly there it's less well developed. You know, Dryad will take that data, it will remove the sensitive data. How does someone get access if they can show they really need it? That's a lot trickier. So then biosecurity, you know, we've dealt with mainly in wet experiments so far. I mean, through COVID, through, you know, H5N1 is the big one everyone will remember. And there we're looking at, for those kind of experiments, you know, was there an IRB? Was their work evaluated for DIRC? Most of the time, if it really was evaluated and really was considered DIRC, it doesn't come to a public journal. So what we're looking at normally are much more borderline cases. So if they've, you know, so we'd still be looking at, has it been through your institutional board? Has it been through, and if necessary, where we're seeing real risk, we'll consult with experts to try and figure this out. But even on the experimental side, that's kind of, we've got that in place for, okay, how do we decide this is DIRC or not? But that if we decide it's tending towards DIRC, what do we do? There's not really a mechanism in place, okay, you can publish this part and not that part, or you can publish it, but leave this restricted. So that's, and that is becoming even, it's becoming highlighted more in the time of AI, when people are coming with more, oh, well, this could be used. It's clearly not DIRC, because as Linda pointed out, but there are risks, and how do we deal with those risks? And so what I can say is, as journals, we don't really have a good general solution. There are case by case that authors are working out solutions, and you will hear about some of those in the case studies. But I think it points to the need for this meeting. Thank you.
All right. Well, thank you, Vala and Philium, for that. And this starts a Q&A session now. So if I'm reading the clock right, those were a well-oiled set of presentations that put us ahead of schedule, but that leaves us a little bit more time for the Q&A, which is fantastic. So please, if you're online, if you have questions, put them into Slido. If you don't have questions, you can still go on to Slido and upvote the things that the questions that you think should be asked to participants. And then just let me ask the speakers to remind you to be brief. I want to start, though, with a question to the group. So there are a lot of ways, as we've seen from some of the presentations, that computational systems can have dual use potential. And one of the main questions that we face then is, when does this computational, or this dual use potential, what's the threshold for dual use potential moving into dual use research of concern? And so I wonder whether the panelists would be willing to revisit that question and speak specifically, what guidance do you have, or what thoughts do you have about where that threshold exists between saying, yes, this tool could be abused, versus this rises to the level now of dual use research of concern. Would you like to go first, Gigi?
I mean, I think the fact that this question is asked shows some of the complexity involved in being able to frame it on and have guidance that can apply to more than one question. The fact that this is so often become a case-by-case basis issue it makes it very challenging to have a broad-based policy, and it requires a lot of expertise to think about it. I would say that, you know, we are not, much of the time I'm thinking about this in terms of more wet lab experiments, but a lot of it requires being able to predict things that are not predictable. And so that is, and that can, because people are very creative in all kinds of ways, you know, your predictions may not turn out to be what is the reality. So, and it's very hard to go back and change something, especially if there are like laws in place. So I think it just reflects the complexity of the question. That's my way of passing that problem to somebody else to answer more.
Okay, would any of the other panelists like to weigh in? Okay, so another question then is whether there are strategies through the training data, model validation, or that are sort of in maybe more endogenous to these systems that could reduce the biosecurity to computational systems that could reduce biosecurity risks that might arise from their dissemination. So I don't know if any of the panelists would like to take that on. Linda?
Yeah, I'll take on one example that's out there at the moment, which is the EVO2 model, which is a design model where they deliberately withheld the viral protein structures in an attempt to detune it in that kind of arena for applications in to viruses. So couple of problems in thinking about that is, of course, if you want to then use that model to do something good with it, like make a medical countermeasure, you now have a model that's actually not as good as it could be for for a important biosecurity purpose. And then the second is how easy is it to then patch it or refine it by retraining it on that if it's open source, right? You can just retrain that model on the or fine tune that model again on viral structures. So and sequences. So I think that it's one of those things, but it seems like a nice thing in theory, but in practice, if somebody really wants to do something bad with it, they could easily kind of do something bad with it. So, or not, I mean, fairly easily kind of recover the deficit. So I think these things seem nice on paper, but I think in reality and in practicality, I'm just not sure how useful they are.
Great. So we have Gerald Epstein.
Thank you. This has been really interesting. It seems that part of the challenge is we've had a certain set of problems and a certain set of tools we've been muddling along with, and I think not terribly well all along. And now we're envisaging maybe this is a break point. We have to think about things quite differently, which is why I was really intrigued by Professor Del Val's hierarchy of the different categories of problems. And I'm wondering, it seems to me a linear problem is one, maybe continue as you've been. We have a problem, you kind of throw the tools we have, the problem gets a little worse, we throw a couple more of the same things at it, we can kind of muddle by. But some of your other categories are telling us that's not going to work. And I was wondering if you could establish timelines. Have we always had exponential problems like this, or is it really a break point in time now that we're facing some of your categories 2, 3, and 4?
I think this has always existed. I mean, if you just explore some, mathematical theory where you have very nice dynamics, and then you can quickly go into chaotic environments, and so this has always existed. I think that the thing that I kind of wanted to show with that framework, with going from linear, exponential, and all the way to discontinuous, is that we need to also somehow anticipate the next step. And we tend to be very reactive instead of proactive. And so hopefully by always having the discontinuous phase in mind, that it's a wild card and we don't really know where it's going to go. Maybe that can help us develop I don't know, safeguards that could hopefully get to that point. But as Linda was mentioning, like it's very hard to predict what that next step is going to be. But that was the motivation for those stages. But yeah, I think they're always existed.
Great, thank you. So there's a Slido question for Philim. Are the 2% of cases in which the code was not published at PLOS Computational Biology consistent with Valda's exceptions, or are there additional categories that you've encountered?
Yeah, they're very consistent, and we have very similar policies. Like, for example, we don't view IP or commercialization as valid reason not to share code, and instead it's more these Percy and/or Dirk or other related reasons. So very similar reasons. Yeah.
Great. This from Slido for Dr. Stewart. Given that wet lab validation of molecular designs is still necessary, do most wet labs have their own on-site modeling cores, or are the wet labs non-overlapping within Silico labs? And I suppose that a kind of follow-on addition to this that has come up is about the relationship more generally between in silico and wet labs. Like if wet labs produce a database, would that be a kind of in silico product then that might require evaluation?
So I think it's worth thinking about that both ways. So, historically, I think there weren't that many computational labs that actually had wet lab capability. The Institute of Protein Design was one, but in fact, DeepMind didn't have a big wet lab, on-site wet lab pairing. So historically, I think it wasn't that common. But I think what will become more common is that more wet labs will start adopting the tools, because the tools are now really kind of facile. Even an idiot like myself can use a by-design tool nowadays. So I think that we will find more the approach being more commonly applied in people who have wet lab capability. I think the generation of the wet lab data, but I think for it to be really, really effective, it is best if they are paired together, right? So the computational folk are paired with the wet lab folk. I think probably a bigger question for this group, right, is if a computational lab produces a bunch of potential sequences and then they get published, maybe that lab isn't going to do anything bad with them, but there could be a wet lab somewhere that is, for some reason, I have no idea why anybody would do something so boring, but would want to pick up all of those designs and then express them and test them in some other place. So there is a theoretical risk, I think, of a lab doing something good, doing something in design, and then somebody else out there in the ether reading that and doing something concerning. But it's not that common that the people have both capabilities at once. But I think it will become increasingly.
Would any of the panelists want to weigh in on that? Okay, so I think one of the Slido questions asks something that's sort of in the background of a lot of people's minds. So the question here is, where are we in terms of designing viruses to be more lethal or tissue specific? And I think that this question basically is asking, where are we in terms of being able to do things that would satisfy one of the existing DIRT categories, like increased host range or pathogenicity or something like that? So I think that's a good question, like where do we stand in terms of that technical capability? And maybe also just being able to do that in silico itself sufficient to make it DIRT, or does it have to be the transition of that to a wet lab be, Dirk?
So I know Linda will have a lot to say about this because it was the focus of our National Academies Committee in our report that just we had our publicly released yesterday. But I'll just start by saying that it's really, I mean, if only we had that amount of knowledge to be able to predict the pathogenicity of viruses by just looking at their sequence. And my favorite example that I use when talking about this to people who don't understand, we have made so much progress over the last decades when it comes to understanding viruses. They think, well, what do you mean? There's like a lot more that we need to understand. Remember when Omicron came around and everybody was like, oh, that clearly is bad. Why did they think it was bad? Because it was so different. It was different in its sequence, and it was like, well, that can't be good. But that's like the limit of how much we understood. I mean, there's people could guess a little bit, but the reality was we had to wait to see what the impact was going to be on hospitals and on, and we had to just wait and see what the clinical effect would be, because we can't predict, even with by that point, pretty well studied, but, still in the dark, compared to influenza, virus. And when it comes to influenza, we are still seeing lots of things that we just don't understand. People ask me like, well, what's the clock? Like what is the mutation that we're going to see that's going to make it go? And like, well, I don't know, we'll know it when we see it, and hopefully we won't see it. But as far as, what is going to be that sequence that makes it from like, this, please do not let your cats outside, do not feed raw, milk or any food to your cats, and do not play with, allow your dog to pick up a migratory bird that's dead, all these things that we are telling people, because we don't want people to get sick. The risk of pandemic is there, but we can't quantify it. And so that's something that, if we had that ability to be able to say, well, okay, let's make a more pathogenic flu, there's some things you can guess that would do so. But as far as that perfect knowledge, we don't have it.
So maybe I'll add a couple of things. So first of all, I think, if it's designing a simple protein molecule like a non-replicating toxin, we are able to do quite complicated, to do that. It still requires a lot of experimentation and so on, but that is something that is within the realm of the possible. For the stuff that, like just pressing a button and getting a really bad version of a virus, I think to Gigi's point, we don't have the data to train the model. And so one thing I think the community should think about, right, is if people are generating vast amounts of data on something, then suddenly the models become feasible or become possible. But even within the realm of COVID, where we had we knew more about that virus than anything else. Even that amount of biological data is not the same level, it's orders of magnitude less data than, for instance, there are pictures of cats on the internet. So our ability to actually train robust biological models is, we're orders of magnitude less we're just not there with the data. But we can, I think, anticipate that the data will get generated by people because having those models would be useful. So I think looking and watching for what data is emerging in what areas might be useful. And so, for instance, models, a lot of data on tropism may be an important thing to track, because maybe that would get us to something that would tell us about host range. But at this point, I don't think we have the data to train the model.
Right. I think those were important comments about maybe the degree to which public perceptions have gotten out in front of what the actual technical capabilities are at this point. So I think that's an important grounding. We have a hand raised in the room. So Roger, would you like to ask your question?
Sure. This is for Linda Stewart and continues from your talk. Dr. Stewart, I wonder if the framework you presented in a slide in which the input of AI was most important upstream might have already been overtaken by events. I'm astounded multiple times weekly by how well the large language models, the frontier models, understand structure. They really, really understand structure, presumably because they've ingested every paper that's ever been published, and the structural biologists have not been remiss at pointing out things about structures that they think are salient, that they think are important. So LLMs help with design at points downstream of planning an initial experiment. They help in the lab as you try to execute your wet lab stuff. So that's kind of in praise of LLMs. When and then the last point is that and it's obvious, but a design tool doesn't need to be 100% accurate, so that you don't need to do any wet lab experimentation in order for some change in state. A design tool that generates 10 designs, one or more of which have a 90, with a 90% chance that one or more of which has the desired function, is good enough. And so that capability, you work in this space, I don't, but in some senses, that capability, I believe, already exists. Maybe that's 90% chance for 1 in 100.
So let's let Linda have a chance now to answer the question.
Yeah, so I think there are two questions there. One question is around kind of agents and how agents might help people in the wet lab downstream and LLMs downstream. That wasn't your first part of that question.
No, not agents. You just use the LLM at many points in the process.
Mm-hmm. So, I mean, I think yes, we are going to see increase in capability in the wet lab from AI, but you still have to have a wet lab, right? You still have to have an experimental system. So there is a limit that is a physical footprint of what you can do. So somebody in their basement, unless they've set up a wet lab, is not going to be able to suddenly be enabled by an AI in that space. I think to your point around what happens in the upstream, in the design phase, I think you're absolutely right. Actually, the critical thing is not how many designs you can generate, but actually how effectively you can filter them. So if you can filter them, if you have really strong filters, then actually the amount of design, then the amount of wet lab you have to do is less. So I think personally that in this sort of if-then framing, I think looking for improved filters is actually really important, because if, say hypothetically, we had a filter that was 99% able to filter out for designs of viruses that were more infectious or something like that, then things would be concerning, right? But if your filters are useless, then you're just putting in a lot of hypotheses, but you have no way of sifting through those hypotheses. So I think the filter is actually a very, very important place to consider when looking at uplifting capability, because the better the filter, the less wet lab you have to do.
So I'd like to add a little bit to that, because when it comes to pathogenicity, it's not just the pathogen, it's the host. And the host is a really critical part of this that is going to be different depending on where different hosts are going to have different reactions to the pathogen. And we are seeing this play out with H5N1 right now with, not to keep bringing up cats, but it's really important to protect them. But the cats H5N1 is a neurological disease that's really lethal to them. But yet it seems like it's not as lethal to people who, it's definitely not as lethal for people who are getting exposed from dairy cows. So is that the route of exposure? Is that something with something inherent in the virus? Is that because the people who are doing the milking are younger, versus, maybe if this is in an older population, it would be different. So, I mean, there's just big areas that we just do not know. And so that data is not in anywhere for an LLM to harvest.
So I want to also invite the people who joined us in the overflow room to weigh in with questions if they have them, as well as other people in this room. I want to turn to, we have just about 4 minutes left. So there's a Slido question that I think maybe different stakeholders here can weigh in on. So it's about who's the adversary that we're imagining when we consider whether tools or information that we've generated pose a biosecurity risk. So is it like state actors? Is it lone wolf scientists, or people in their garage? And it is the concern that, because one concern you could have is if someone in a lab does something and that escapes, or there's an accident, but you're not worried about lab escape of in silico work. So what's the pathway then? You're worried about what, lone wolf actors, state actors? How does that influence the way we think about these questions?
So the basis of the report that Linda chaired is that was the 2018 National Academy's report that developed a framework for how you can think about these risks. I think that you have to walk through every problem with that in mind. Not just which actor you might be concerned about, what barriers they might face in misusing your technology, but also what kind of resources they have, what kinds of special equipment they might need, what is the motivation that they could use, or what would be the weaponization potential for whatever it is that you're concerned about. I think there's a whole suite of concerns that you need to walk through, and we can systematize those concerns that you need to walk through, but you have to walk through them at that point in time with whatever technology you're interested in assessing.
I don't know, Philip or Vala, if when you think about the review of materials that you receive, does the threat actor influence the review process?
I mean, a little bit, I guess. There is a consideration of state actors, and certainly depending on geopolitics, which country the reviewers are in who are going to. So if you're really thinking about Dirk, then you would have to be thinking about, who do I ask? But yeah, for the most part, we don't want to be the ones thinking about sort of who we're protecting against necessarily. I mean, I think there's also the other side of it, which is the benefits and sort of allowing access to the benefits. So you also have the risk of preventing access to the benefits. And if you kind of get into the medical sphere, as sort of Sarah was talking about, and you start doing sort of epidemiological modeling, then you're starting to get into realms where your actors could be insurance companies or people who want, sort of, there could be financial rewards and risks.
Yeah, I definitely think we think about who the potential actors are, because you want to sort of create a theory of why there might be a concern. You don't necessarily need to be exhaustive, because one is enough, right, to sort of cause a concern. And I just want to reiterate something that Paula brought up earlier, which is, to go back to this, like, talking about 2% or a small number of the, percentage of papers or dissemination products that might be subject to this, is part of that is because it's not the majority of what we see, then we need to reach out to experts to sort of talk about this. It's not simply us sitting there thinking, is this a problem? We need to engage other people, other experts to evaluate, rely on the reviewers, editors, empower them to sort of raise flags and things like that, and make sure that these things get evaluated. So it's not simply us in a room thinking about it, but reach out to other people to find out what we need to do.
Well, I want to thank all of our speakers for this session, really helping us frame the meeting and prime the pump and the questions from the group. I know that there were other questions in the room and on Slido. There'll be more time for those things. So we're going to now transition to session 2, which is moderated by my colleague Jassy Panu. So Jassy, over to you.
Right, thank you, Alex. So I've got my camera, got my mic on. Hi, everyone. My name is Jassy, and I'm a postdoctoral scholar at Stanford University and a fellow at the Johns Hopkins Center for Health Security. So now we'll be moving on to session 2. The goal of session 2 is to discuss the benefits and risks of dissemination, so of studies, models, and tools involving computational approaches. We have some experts joining us today to illustrate these theoretical discussions through real-world examples. And we hope that these examples will highlight the complexity of biological systems, particularly when involving those computational approaches, and also showcase the broad range of dissemination options that either do or should exist. So our 4 excellent speakers will present their research in different domains of the life sciences, using advanced computational models and their broad applications. And they'll also help us review biosecurity considerations at different levels, including at the funder level and dissemination level. So as a reminder, all of our speakers' bios can be found on our website, and we'll put that link in the chat again. Each of our speakers will have 7 minutes maximum to present their work, and this will just ensure that we have enough time for a rich Q&A with audience participation after the speakers present. So I'll move ahead to our first speaker, who is James Diggins. James is head of biosecurity for Twist Bioscience, as well as the chair of the International Gene Synthesis Consortium. James, you can go ahead and turn on your camera, unmute yourself, and also share your slides. I will stop you at 7 minutes, and the audience is invited to submit questions or upload their questions into Slido during the presentation. Over to you, James.
Thank you, Jesse. And I appreciate the opportunity to speak to you all today. I'm going to talk through an example from the world of DNA synthesis screening. If you're not familiar with Twist, we're a company that makes DNA. We have headquarters in San Francisco, manufacturing facilities in California and Oregon. We're a global company. The short version, since I only have 7 minutes, is we make a ton of DNA. And I think as we've already kind of heard this morning, you can think of DNA as a dual-use technology. And so when these technologies were coming together kind of in the mid-2000s for being able to make very long DNA sequences, gene length, there was a lot of work that came together both from governments and industry to figure out kind of best practices about how to minimize the risk of misuse of these technologies. And you heard, I think, Linda talking this morning about kind of the difference between design and then bringing a design into the world physically. DNA kind of sits at that boundary. And so most of the large DNA synthesis companies in the world screen their orders to make sure that those orders will be used responsibly. That right now looks like customer screening, making sure we know who we're making something for, as well as sequence screening. So making sure we understand what we're being asked to make. That sequence screening right now is very focused on homology-based screening. So we're essentially saying, given the thing you've asked us to make, what is it similar to from the natural world? And can we make any judgments of its biological function based on those similarities? The work that has kind of started in 2010 till now has largely been focused around an organization called the International Gene Synthesis Consortium. That's a group of about 40 companies that are working together to kind of build these best practices about how to do these things in a way that minimizes the risk of misuse. I'm lucky enough to chair that organization. And we, as synthesis companies, have kind of watched AI biodesign tools grow in capability to the point where they really now can reduce homology to naturally occurring proteins kind of while maintaining the function of those proteins. And so into this mix of kind of historical practices and new disruptive technologies, Microsoft, the office of the chief scientist there, Eric Horvitz, who has been an amazing partner in this work, approached IGSC companies and said, we think these technologies probably create a vulnerability in your screening systems. Can we work together on that problem? And so Microsoft and the IGSC, including 4 companies inside the IGSC that make screening tools, so computer systems that are estimating risk associated with incoming sequence orders, teamed up and engaged in kind of a year-long project to do a few things. So first is to determine, is this actually a vulnerability? So Microsoft carried out this pilot project that showed, yes, if you use AI tools to engineer proteins, you can change the primary amino acid sequence sufficiently that you can get past some of these screening systems. And from there on, that team, we first agreed to follow this CERT vulnerability disclosure process that comes from the cybersecurity world. Microsoft built a really sort of state-of-the-art protein reformulation pipeline so we could kind of scale up this kind of testing. That group worked together to kind of assess that vulnerability, develop mitigations. And then once we had mitigated this risk, we wrote this up as a publication because we really wanted to share the fact that this work had occurred and here's kind of the state of the art and where things sat. And as part of this kind of responsible disclosure, we submitted that paper before we submitted it to any journals to a bunch of experts across several fields for kind of pre-publication review, just to get a sense of did these people who work in these domains think that there was any kind of information hazard represented in that paper. And what I will say, though, I think we learned in that process was that those reviewers had incredibly broadly divergent opinions. So it ranged all over the map from, you can't tell people the name of the tools you were using for this, all the way down to like what you have is totally fine, but you should add more content, right? It was completely divergent in terms of the degree to which people who are experts in these fields. So these are not average Joe public. These are people who really know what they're doing. They still had very different tolerances for risk. And I think and I can't speak for my co-authors, but what I will say I took away from this process was that often the least risk tolerant reviewers will end up having an outsized impact. And so your publications kind of end up removing information until all the reviewers are satisfied. And you kind of get to this kind of, I don't want to call it lowest common denominator, but removing more information than maybe many of the authors might have thought was necessary just to make sure that all of this pre-publication review kind of was satisfied. And I think to me, this pointed out that we need better vocabulary and categorization around these kinds of information hazards, because much of the time I feel like we were talking past each other. So, there's a difference between like, this is a thing you can get via trivial web search versus this is a thing no one in the world knows except these, 2 or 3 individuals. And there's a huge spectrum of difference between all those things. And so I think what this really pointed out to me was that we need more formal norms around how we draw lines of what is and isn't a significant information hazard. And importantly, we need structured, constructive ways of defending those lines. So when we were having these publication reviews, if I felt someone, was wrong, it was really my opinion versus this other individual. And like, that is not the kind of constructive disagreement you want to have. You want to be able to refer back to some kind of framework and see if you can categorize these things in a more structured way and then argue about where those lines should be between those categories. And I think we just really encountered the lack of that kind of more structured way of thinking about these. But I really, again, want to thank the team who engaged in this work. I think it was a really important paper. But I think it really did point out the need for this more highly structured way of talking about information hazards. And so I will leave it there.
Great. Thank you, James, for that very interesting overview of synthesis screening and of your recent work. Our second speaker is Anthony Gitter, who's an associate professor at the University of Wisconsin-Madison. Anthony will review his work on protein design, specifically dynamic models of signaling pathways and transcriptional regulatory networks from high throughput proteomic and transcriptomic data. So I'll hand it over to Anthony. Thank you.
Okay. We're doing the remote share. I'll replace it with mine if that's okay. Okay, I am in the driver's seat. Great, so I'm not actually going to talk about my own research in protein machine learning today. I want to really focus on the theme of this workshop. I'm very glad that Linda introduced this excellent report because I don't know about all of you, but it came out less than 3 weeks ago. It's 128 pages long, so that's a pretty intense reading agenda if you wanted to get through it all. I did try to read it all because it's excellent. So there's a lot of great content in here I might refer back to later, and I'll try to give context when I refer to it. But it really digs into benefits and risks of AI. So I'm going to try to stay laser focused on dissemination of AI models today and not talk about my many opinions on the risks. We can discuss those later if we would like. So I'm thinking primarily about dissemination processes and what that means. And I thought the best starting point was actually coming from this great paper from someone in the room, Richard Sever, who had this 2023 viewpoint of what the current publication process looks like and what it might look like in the future. And we have a somewhat traditional model on the right side, which is publisher driven. So here we might have multiple journals in a publisher family. And this is a classic model where a manuscript is submitted, it stays within a closed review process, and it eventually emerges and is publicly shared at the end of that process in some journal within that publisher's family. And maybe it cascades down some prestige tier as it's drawn here. And I think if we imagine that publication has to do with the biological AI model, it could be the case that the AI model, the code, the data is not released publicly and disseminated until that process is completed. Reviewers might have access, there may be some private sharing, exchange back and forth, but the release may not happen until the end. And in a contrasting view, this open ecosystem, we could have multiple preprint servers, ChemArchive, BioArchive, MedArchive, Archive, the good old archive. And then a lot of things happen downstream. So you can have peer review, things that are managed by the scientific community. There's still a need for content verification. So something that's managed at more of an editorial level. So that role doesn't go away. You still need someone to look at image manipulation and plagiarism and things that are not scientific peer review. But what I find interesting in dissemination is that AI model release can happen anywhere, anytime, and we've seen this already. It can happen before the manuscript hits the preprint servers. It can happen alongside the preprint with the first version of a preprint. It can happen later because some people don't like to make their code available until it comes out in a journal. And this is a author developer driven process. Anything goes, and the developers of the AI tools are controlling that process, not the publishers. I think that's important for us to grapple with here today. And also, there's a lot of people developing these tools that don't care about publications and have completely sidestepped this entire process. Some of them are in industry, some of them are doing their own thing. Some of them, like Arcadia Science, have self-publishing platforms and have said, we're doing things differently. There's also a lot of blog-based dissemination going on. Some of that because it's industry-driven. I picked an example from one of our future speakers. They may speak to this from evolutionary scale. Sometimes we have interesting examples where the couple days it takes to screen a preprint is too slow. So people need to get the word out right away and get a manuscript up and their models out, and then the preprint comes out. And you also have this emergence of, I guess, what we might call like the garage startup culture. I was surprised by a blog post of somebody working on what my research group does. And in the Twitter thread, it was described as a fun employment project. And this was somebody fine-tuning models, doing things that my academic research group does, and they share the code and model weights and write a blog post about it. So this is very much outside the let's publish a manuscript in one of those pipelines. So it's all developer driven. That's the punchline. I think we've hit on a lot of these things, but I'd want to write them down. What is going in a AI release? So there's definitely ways to distribute software. Sometimes that's source code. There's common platforms to distribute code, but you can stick code anywhere. It's usually small in terms of file sizes. It could be executables. Maybe that's popular in industry if you don't want to share source. If you want to give access to software, but not provide the actual code, that could be distributed through internet connectivity, through browser-based, through a web server, ways to connect to it programmatically. You can have web services like maybe an Amazon service that lets clients, customers pay for your service. There's containers, things like that. Data sets can be distributed in many different ways. Sometimes there's no new data used, so it may just be pointers to public data sets that exist or reprocessed forms of those. Hugging Face became popular first in the general AI community. Now it's popular in the biological AI community because it works well for distributing data sets. There are good generalist AI or data set repositories like Zenodo, Figshare that are good for archival, minting DOIs. So those can be used for data. The list goes on. There are also things that I wouldn't recommend, but we see them. Google Drive, there's no permanence. Access is poorly managed, but you see everything and anything. Same thing with model weights. Model weights are essentially the rules that tell an AI model how to make predictions, but when you save them, they become files. So those files can, again, get stuck anywhere and everywhere. There are better places to stick them. There are best practices that scientists would recommend, but they get stuck everywhere and anywhere. Even torrents, which is really crazy. So we need to think about widespread dissemination options. We've already hit on the benefits of open science, so I'll actually skip over this and move on to something that I've been really trying to advocate for with a group of colleagues that I think as a community we should think about open science by default unless there's very, very specific needs to have controlled access. And then we should think about what the best options for controlled access are. In some cases, if the harms so greatly outweigh risks, we may not want to do the research, but we need researchers, developers to think about that far upstream. And this mixing of IP interests, commercial interests, and biosecurity in the publication process has been very detrimental to biosecurity risks. And I can give specific examples later. And lastly, I'll just point out, there are some repositories already that control access. Some are better, some are worse. I've started talking to local librarians. They think a lot about these things. We have experts that know what are good ways to control access. And we can try to hit on what are some of those desired features, what existing repositories offer those features, perhaps besides reinventing the wheel. So I'm a bit over, I'll stop there.
Excellent. Thank you, Anthony, for sharing your very helpful insights regarding dissemination options. Our third speaker is Nicholas Safranu, who leads the Frontier Intelligence team at Evolutionary Scale. Nick will discuss his work on protein evolution simulation and new protein designs, as well as Evolutionary Scale's responsible development framework. So Nick, go ahead. I can see you turned on your camera, unmute yourself, and my understanding is you have no slides, so go right ahead.
Great, thanks very much. So, at Evolutionary Scale, our mission is to develop artificial intelligence to understand biology for the benefit of human health and society. And we want to do that in ways that are open, safe, and responsible, and in partnership with the scientific community. And so I'll just give a little bit of context on the company and our work for those that aren't familiar with it. Over the past 5 years, we've developed a series of protein language models that are widely used across life sciences. One of our earlier model families, ESM2, has been cited sort of thousands of times by researchers for tasks like variant effect prediction, protein structure modeling, antibody engineering, drug target discovery, things like that. ESM2 also powers ESM-Fold, a fast and accurate structure predictor. And these tools are open source. They're downloaded millions of times per month, and they're broadly integrated into academic and industrial workflows. And as a company, we really have values around openness and responsible development and deployment. So more recently, we've introduced two sort of newer models, ESM3 and ESMC. And today I'm going to tell you about the steps that we took during the release and deployment of these models. So ESM3, it's a multimodal generative model that can jointly reason over protein sequence, structure, and function. It can follow complex prompts, generate novel proteins far outside of known evolutionary space. ESMC, it's a representation-only model, kind of a successor to ESM2. And ESM3, it exceeded the compute thresholds outlined in the previous White House executive order on AI. And so, as part of its release, we also followed associated reporting requirements then. So to guide our development and deployment decisions, we built a responsible development framework at Evolutionary Scale, and it rests on three pillars. The first one is that we will communicate the benefits and risks of our research. The second one is that we will proactively and rigorously evaluate model risks with the scientific community. And then the third one is that where warranted by model capabilities, we will adopt mitigation strategies and precautionary guardrails. And we really think about risk in context. So that is, relative to existing tools, laboratory practices, current safety infrastructure. And we ask questions around whether a model meaningfully lowers the barrier to harm for users without advanced expertise. And importantly, we also weigh these risks against the major scientific and public health benefits that can result from releasing models in a way that makes them widely accessible to the scientific community. So, more concretely, the first thing we do is perform a series of internal evaluations. We sort of came up with a set of risk-relevant tasks. These can include things like sequence understanding, fitness prediction, sort of structure condition design for areas of potential concern, such as viral proteins or sort of select agents. And we can compare models with and without various mitigations, such as data that gets included or excluded from model training. The second thing we do is we conduct a sort of structured expert review. External reviewers are provided with the detailed internal evaluations that we've performed. If appropriate, they're provided with model access. We have a sort of key set of questions that we ask them. Three of these questions are: does the model increase the likelihood of harm? Does it lower the barrier to harmful misuse? And then, do the overall benefits of release outweigh the risks? And so we collect that feedback from experts and review it. And then the third step is that we have sort of defined access policies based then on model capability and risk. And this can include things like open weight release for the sort of lowest risk models. We have the option to do sort of gated API access for sort of higher capability models. For models that are behind APIs, we can do things like input-output filtering for sequences of potential concern. We can also log the sort of sequences that are put into our APIs for sort of audit and oversight. And we can also have methods for vetting partners and then providing elevated access for vetted partners. And one more thing that we can do is a sort of staged release process where we can think about beginning with a controlled API access and then doing a more open release after we've had a chance to learn more. So, very concretely for ESM3, I mentioned a little bit earlier about what that model was. This was published recently in Science, and as part of that process, we wanted to make the models widely available to the scientific community. And so in this process, we actually trained several versions of the model, including different sizes. We see that capabilities change with model size. And we also trained a dedicated open model that was a smaller model And we trained that with precautionary mitigation. So we actually excluded millions of viral sequences and select agents, and we filtered out sort of thousands of pathogen related keywords. And these measures reduce performance on sort of sensitive tasks like viral fitness prediction, but preserve general utility across the protein sciences. Now, I want to say that this decision, came with trade-offs. Many researchers, use models like ESM to study viruses, develop therapeutics like vaccines, and limiting those capabilities, can reduce, reduces the scientific value of the tool for those people. I think in this case, we took a cautious approach, and we wanted to give time for external expert review and community feedback. And I also after structured engagement with the community, we're definitely going to continue to consider the risks and benefits of including sequences like this in future models. I think it's definitely something that the community needs to continue to weigh in. So for this case, all the expert reviewers, they found that the risks and benefits, they looked at the risks and benefits of ESM3 Open and concluded that the benefits significantly outweighed any risks. So, in general, we followed a similar process for ESM C, which is this representation model. There we decided actually to include all sequences, all evolutionary sequences. And again, in this case, the reviewers, found that the benefits of doing that greatly outweighed the risk. So really in closing, I just want to say openness, it's been a central principle of the scientific community throughout history. Yes, there's a rapid pace of AI advancement right now. It has the potential to dramatically accelerate our understanding. And, I think we really think that, these capabilities need to be considered responsibly. And we do think that there is a path for sort of transparent and open sharing of scientific advances if really done, responsibly. So with that, I want to, thank you again.
Great. Thank you, Nick, for that excellent overview of your work at evolutionary scale and also the suite of options, dissemination options that are available to you. So our last speaker is Jamie Yassif, who serves as the Vice President of NTI Global Biological Policy and Programs. Jamie will share her experience working with funders to incentivize scientists to follow biosecurity norms and best practices for in silico research internationally. So Jamie, if you can make sure your camera is on and unmute yourself. And if you have slides, feel free to share your screen.
Great. Well, thanks so much for convening this really important discussion and for the opportunity to participate. So I'm going to be talking about responsible development and dissemination of AI biomodels as part of our work at NTI. And just for context, NTI, we're about to celebrate our 25 years anniversary. We are a nonprofit, nonpartisan global security organization really focused on reducing biological, nuclear, and emerging threats that imperil humanity. And I'm, as the VP for our bio program, I'm responsible for the bio part of that mission. So quick overview of what I'm going to talk about over the next few minutes. I'm going to start with framing some of our work on the Biofunders Compact, how funders think about this work in terms of pre-funding biosecurity review and assessment, and then pivot to some of our thinking about dissemination and the fact that it is a really important consideration for biosecurity. I'm going to talk briefly about a very early stage pilot project that we're looking into on managed or tiered access that I'm working on with my colleague, Sarah Carter, who's here with us, and then share some key takeaways for the committee. So in terms of the Biofunders Compact, what is it and why are we doing it? What's our theory of change? So it is a public commitment by funders of bioscience and biotechnology research and development to incorporate pre-funding biosecurity review into their decision-making. And the theory of change is to reduce the probability that funders will support unduly risky bioscience research and to provide strong incentives for scientists and technologists to adhere to biosecurity and biosafety best practices. Certainly, the US government has been very forward-leaning about doing pre-funding biosafety and biosecurity review. Part of the reason we started this project is that that's not necessarily true across all governments, and there are also a significant and growing role played by private funders in philanthropy and venture capital, and we wanted to help them bring them into the fold here. We did launch the Biofunders Compact last year at the Global Health Security Conference in Australia in June of 2024. We partnered with CEPI, the Coalition for Epidemic Preparedness Innovations, and they co-launched this with us. And they were one of the 3 founding signatories, along with the Global Health Security Fund and Sentinel Bio. I suspect most people around the table are familiar with CEPI, but just for level setting, they are a global organization that's really focused on advancing medical countermeasure development with a focus on the 100 Days Mission and rapid vaccine development. I think they're also interested in therapeutics. Okay. In terms of the content of the commitments, so there are five broad commitments, but basically there's the signatories of this compact and others that we're looking to recruit, they recognize their responsibility as sort of shapers of life science research and development, in addition to advancing innovation and the benefits of science, guarding against deliberate and accidental misuse, and undertaking a number of high-level commitments. So there are 5 here. I'll highlight 2 of them. One is a pre-funding biosecurity and biosafety review as part of decision-making practices within organizations. And then also committing to share best practices for biosafety and biosecurity risk assessment and risk reduction among funders. So, well, I guess one other thing I'll say is that within scope of the Biofunders Compact is both sort of dealing with traditional biosecurity issues, what we as a community have been thinking about for a long time, including dual-use research of concern and enhanced pathogens of pandemic potential, EP3. But we're also thinking about the same questions that this committee is thinking about. How do we safeguard in silico models or AI bio capabilities? The first set of questions I think is more well developed, and it's very early days as we grapple with the latter question. This is meant to be international in perspective and scope, as I mentioned earlier. And we know, for example, as part of the international perspective, that there are some funders who aren't necessarily part of this compact yet, that are in Europe and the UK that are starting to ask prospective grantees who are asking for funds to develop AI models relevant to biology, what kind of provisions are you going to put into place to make sure that your model isn't exploited for deliberate misuse? And so I think the question is on the table. Not all the solutions are there yet, but people are starting to look at that, and I think that's useful. So in terms of, the topic of today's discussion, the Biofunders Compact is very focused on elevating biosafety and biosecurity considerations among funders. And from our perspective, dissemination, the topic here, is a key consideration for biosecurity. Managed access is one mode that we are looking at as a way to handle a subset of models that might be particularly of concern. And we think that can play, the reason that managed access is important and valuable is it can play a really important role in safeguarding tools against misuse. And the particular hypothesis we're working from is not only are we thinking about managed access or different modes of dissemination, but if we want to be building in other kinds of guardrails like filtering inputs or outputs, as we've heard from some of the other speakers, if everything is disseminated fully open source without any constraints, it's very hard to guard against stripping out those guardrails. And so that's an important consideration from our perspective. We also think it's likely that taking these kinds of steps could help build public trust in scientific communities. So in terms of the principles, how we're thinking about it, so I think there are basically 2 principles. One is we want to have balance, as we've heard from other speakers. We want to balance access, including equitable access. Can't see what that says. 2 minutes. Okay. We want to balance equitable access with security, and we think that that's feasible. And in fact, this is already happening. CEPI is establishing managed access frameworks within their work on developing vaccines and therapeutics, including work that deals with dangerous pathogens. And the second point that's really important is tiered frameworks will be essential. So we're not saying all of these tools should fall under a managed access framework. There are a lot of tools that are low risk that will not need managed access, but there are going to be a subset of tools that pose higher risk, or we think that that's likely to happen in the future. And there should be more criteria for access, and we should think about what that looks like. Another point I'll make is that, and this is a point we made in our 2024 report on guardrails for AI biodesign tools, that access is not all open or all closed. There's a spectrum. from, sharing all the code and weights to access via an API and everything in between. And the discussion about varying levels of access is not restricted to this community. It's part of the International AI Safety Report that was recently published. It also sort of talks about that in broader context of AI models and LLMs. There are several key needs for managed access. I'll just hit a couple of high points in the interest of time. So we need to figure out what these tiered frameworks look like, what falls within managed access and what doesn't, and what are the criteria. That's a work in progress. It's going to cost a lot of money to make these kind of frameworks happen in reality, and like independent academic researchers or small startup groups don't have the resources to do this, and so government funders have a really important role in supporting the development and maintenance of these. And I think in order for this to work in practice, this is going to have to be aligned with the dissemination policies of journals and non-traditional funders, as we've been discussing. We need to figure out what that looks like. So key takeaways. So the Biofunders Compact asks funders to consider biosecurity, and dissemination is a key consideration. We need to have balance between biosecurity needs and equitable access, and as well as just basic scientific progress and innovation. We do think it's feasible to strike that balance, but it's going to take work. There are a range of dissemination options. It's not black or white. We can explore that range. And this work will require support from government funders, including the likes of NSF and NIH, and they should support these kinds of tiered access platforms. Thank you.
Great. Thank you, Jamie, for sharing those key takeaways as well as your general experience regarding how funders could approach these important considerations. So I'd like to thank all the speakers for their insightful presentations, and we now will proceed with Q&A. Speakers, as you answer questions, please be brief and direct so we can give you all an opportunity to participate. And we also encourage you to build upon each other's answers. For the audience, you can submit or upvote questions through Slido, and questions, again, that are upvoted will be prioritized. But I wanted to start off with a question to, I think all of you could contribute to this, which is, how did you all decide how to disseminate some of your results and resources that you brought up in your presentations? How did you approach that decision-making process?
I mean, I can lead that off. I think we tended to frame it more in the kind of classical framing of a cybersecurity vulnerability. And so that gave us a lot of existing ways of thinking through what was safe and what wasn't. I think what we ran into was the lack of certainty around how to frame vulnerability or information hazards specific to this kind of domain of computational methods in bio. And so one part of that was really helpful. The other part of it was a challenge.
Can maybe say, I think we generally look first at the scientific community and how, they've approached, sort of advanced technology in the past. There was an effort maybe a year or two ago around sort of responsible biodesign tools, and a sort of community statement that was put together, and a number of the people at Evolutionary Scale were involved in that, and that was quite influential for us when we started this process.
I think our motivation to start to talk about this with the scientific community and more broadly is just recognizing really rapid advances in AI bio capabilities, and the point that it's like hard for governance frameworks to keep up. And so we know that right now, the sky isn't falling. We don't see imminent risk tomorrow, but we do think that on the horizon, we may be seeing it. And we want to get out ahead of it and start to tackle some of the really hard problems and address the gaps that we're seeing where we don't have solutions to some of these problems.
So in my research group, we develop a wide variety of computational models, and part of my thought process is not necessarily, do I think these models are risky, but if we release things of this nature, will others be concerned? And that's usually the entrance to our decision-making process. So if we have models that involve proteins and protein biophysics, I envision that they will be universally viewed as quite low risk and not even dual use. If we are working with viruses of any sort and coming up with new ways to model viral genomes, even if they're prokaryotic viruses, I know that some people will see viral genomes and start to ask questions about that. So that's when we've decided to engage with experts pre-release. So that's usually our thought process before releasing models and deciding whether we should ask questions very similar to what Nick described within evolutionary scale, because we've consulted with some of the same people in this community about how to develop pre-release questionnaires.
Great, thank you. And for the example, this is a bit of a follow-on question. For the examples you discussed, was there guidance that you found that was readily available from, for example, your funders, your institution, the government, other publishers? It sounds like some of you spoke with experts during your process. If you did receive any of that guidance, did any of it align or diverge? I know, James, you touched on this during your talk. But happy for anyone to take the question.
Yeah, I mean, I can say we actually even, we spoke with experts even in the development of our responsible development framework. And then in that conversation, we wanted to make sure we brought in folks that policy experts. I know there are some people in this room and on this call that we spoke to in the development of that framework. And so even at the earliest stage there, and I think in terms of making the framework, it was very, very helpful to get a sort of diversity of perspectives. I will say that maybe in contrast to James, I actually don't think we experienced quite the level of varying opinions there. And so I think it maybe goes to say that maybe the details of how one runs these sorts of expert engagements and maybe that can manifest in some of the differences there.
Go ahead.
I have a question for James, if you could, but anyone on the group can also address it. But I'm curious. So your paper, it doesn't seem like it fits into the traditional categories that we have for DIRC and EPP from physical lab environments. But it also seems like it meets a kind of dual use criteria because it's about how could we evade detection. But it isn't clear that we want to validate the results. So do you actually maintain functionality in the so that the model makes some predictions, but do those predictions play out? So how should we manage the tension here between the potential, the idea here that this could be used for nefarious purposes, with the balancing the scientific need for people to vet what you've done and confirm or evaluate the content of the research?
Yeah, I appreciate the question. And I think that's really exactly what we wrestled with throughout the whole, pre-publication review process, was really how to walk the line between you want to publish something that allows people to understand the work that was done and ideally reproduce it, but you also run the risk of kind of empowering follow-on kinds of efforts to figure out newer vulnerabilities in those systems. And so we kind of wanted to walk a line between those two things. And I think part of the problem is that you really can't ground truth a lot of these. You can't take the modeled proteins that came out and make them in a lab and see if they're excellent weapons, right? That just can't happen. And so it's very difficult to figure out where and when something is really creating a threat or not. And so that leads to this kind of urge to be as vague as possible, which I think on the scientific side is not helpful, because in the future, homology-based screening is going to get less and less useful. We're going to need models to help us defend against models. And what is the process by which we can make those models if we're all unable to really speak openly about what the development process looks like? And I think I don't have a good answer to that. I just think it's a really difficult problem.
Yes, I very much agree with that, both on the models defending against other models, and also I think the risks of kind of creating roadmaps to harm by being maybe putting too much resources into say actually developing these proteins. I mean, we very much think about sort of performance on peaceful proxy tasks. I mean, you could say our example of making a GFP that is 57% sequence identity to known GFPs but retains its functional fluorescence is maybe a relevant data point in this conversation, but is a very much a peaceful proxy task. And so I think particularly when things are hitting the real world, hitting the actual physical world, I just think that's very important to take into consideration.
Richard, did you want to?
Yeah, I had a clarification request from James, really, because it was noticeable that, Nick, you said that you didn't see this divergence amongst the opinion of the people you consulted. And James, you said you did, and you referred to it as a difference in risk tolerance. Did you mean risk tolerance, or did you mean assessment of risk magnitude? And does that make a difference?
I would say there probably was a diversity of both of those things, in the sense that some people would see some aspect of the paper as posing, some level of risk X. Some other party might see the same level of risk. One party was more scared of that risk than others, right? Or they may have estimated the risk to have been different. So I think both of those axes were at play, but it pointed to the fact that like a lot of that risk assessment wasn't grounded in truth, or even I think, as we were just talking in response to Alex's question, in the ability to even establish ground truth in terms of like what are these systems spitting out, and are they real threats? And so in the absence of that ground, being able to ground that, I think people were prone to kind of imagining scenarios, and depending on how imaginative those scenarios could be, they would kind of amp up their risk assessment and tolerance in line with that imagining. So it was just a process by which there wasn't a lot of ground truth, and that made it difficult.
I will say we really encouraged our reviewers to focus on concrete risks and a little bit less the sort of hypothetical imagined future, because I think things can sort of wildly diverge at that point, where if we think about the risk in the context of today's tools and the things that are sort of immediate and grounded, maybe that was an area there.
All right, there are some quite popular questions on Slido that I wanted to surface. So the top upvoted question is that the workshop was framed or is framed around computational approaches broadly, but our discussion so far today has focused on AI models. So the question is, are there frameworks for assessing non-AI computational modeling and bioinformatics tools? And do you all think that such tools should be included in this conversation? And if so, why or why not?
I mean, I would certainly say I think of this as an abstract collection of capabilities, not necessarily tied to AI. And the same questions around what is an information hazard and what isn't persist for me. So I don't see them necessarily as a distinction.
Yeah, I don't know if it's helpful to draw any hard boundaries, but I think the reason that there's been increased discussions around machine learning and AI, and the reason why we're having meetings like this, and the reasons why we're seeing progress in certain computational scientific fields, is because machine learning has driven certain successes, as the other NASEM report has highlighted, that has given us new tools that work better than a certain class of other bioinformatic methods. So that's my understanding of why a lot of the discussion focuses on AI. But there's no reason to exclude all other bioinformatics methods necessarily if some of them happen to meet dual use research of concern classifications, and some of them likely do. I think they also can be important to include in some of these broader discussions, because some of those tools can produce the same outputs as our latest and greatest AI tools, like things like variant effect predictors, and it can be useful controls in a sense. So if a 30-year-old bioinformatics tool that's based on pure evolutionary sequence conservation performs as well as some task as a 98 billion parameter large language model from evolutionary scale at predicting some viral phenotype, that should tell us something about where the state of the art in pandemic-related viral phenotypes might be. So they should be considered, even if we're not necessarily concerned about dual use.
I mean, just a short intervention to answer your basic question. I'm not aware of any frameworks for evaluating risks posed by in silico models. I agree with my co-panelists that there doesn't necessarily need to be a meaningful distinction, and you could certainly encompass the broad suite of tools. I think it's really about the performance and the like what is the outcome and the end state, and how much is it in like uplift capabilities for a broad range of actors?
And we have a comment from the room. I'll ask you to speak up so that the
Yeah,
mic
no,
picks you up.
thanks so much. I'm Tommy from Johns Hopkins, and just a comment and a couple of thanks. Sure,
Please.
unless you move that forward, I'm not sure.
Let's do that. Let's do that.
Just maybe get the panelists to react to a couple of possibilities here or statements. First of all, I agree completely with the sentiment Anthony expressed that the default position here should be open science unless serious biosecurity risks, the most serious biosecurity risks are identified in the process of evaluation or risk assessment. And in my view, those would be risks that substantially increase some kind of pandemic outcome. The second comment was just about the work of this committee being looking forward and not just about this moment in time. I think Linda made it clear that filters are going to get better, data is going to get better, these models are going to get better collectively. So our job, I think, is to talk about governance and publication for when those things cross the line and become highly consequential. So even if we all agree, I doubt we all agree, but if we agree that the risks are not present today, I think enough of us have said that the risks are going to change possibly quite rapidly. And if this community doesn't take control of this conversation and act highly responsibly, it could lose control of the conversation. I think that's just my view. I don't know if others would agree with that. The next thing I'd say is that I think it is worth questioning whether we need to accept the most highly dangerous outcomes of these models in order to get the benefits of the models. I think in the wet lab experience, I think we should challenge the notion that we have to do the most highly consequential and risky research in order to get the outcomes of virology research or new vaccines or medicines. No vaccine manufacturer has said before, I need to make a highly dangerous virus in order to make this vaccine. So I challenge that notion in silico as well. And I think it's quite inaccurate to say that if we'd done more PEPP research or gain of function research, we'd be better positioned with H5N1 right now. And then lastly, to Roger's point, I think if an AI biomodel gets to the point where we can say there's some % chance that this will create a very dangerous product, there's only a 50% chance of that. The outcome should not be, let's go to the lab and see if it does. That's enough. 50% is enough if we've created a new strain that is more consequential than the most dangerous existing viruses on Earth. So I don't think we should pursue that. I think that's a strange assumption that we're going to have to test everything out in labs. I think we should be questioning that assumption. And happy to get reactions to that. I think what we saw from evolutionary scale is very impressive and shows that we should kind of reject this idea that everything is unanticipatable. We can't judge anything in the future. I think evolutionary scale is doing as good a job as we could do right now in risk assessment. And I think risk assessment should be a default position for people who are working on the edge with AI bio. It should not be the default position that we can't judge anything or can't anticipate any outcomes. So anyway, I just wanted to commend a lot of the responsible work that I see going on, and a lot of the responsible comments that are being made.
Right. We'll have reactions from the panelists, and I think we also have a reaction from the room. So whoever would like to go first.
Maybe I'll just say, yes, I very much agree with a lot of the sentiments, that particularly this questioning this need to synthesize things, that we have a high prediction that would lead to harm. I agree. We really don't want to create those roadmaps to harm and roadmaps to misuse. I think there's no need to bridge that in silico to physical divide in that context. And I think we should also, I think openness is a fundamental tenet of sort of how science advances. And I think it's important that we do these risk assessments. But I think even things like managed access, the approaches we've taken, I think that needs to be reserved for the cases where we're most confident there is like real world risk. So I very much agree with those sentiments. Thanks for having me.
All right. Excellent. I think in terms of comments, I saw Hector, then Roger, then Valda, and I see we have a few more. We have a few more minutes, so we'll just try and keep them brief so that we can get through everyone.
So I would say, I don't know if you agree, that one of the biggest problems we have is that we can't predict the functions of these proteins, right? And I think one of the ways is to actually do those experiments that can be, look risky to you, to actually train models that can be used later on to avoid it. That doesn't mean that it has to be done, in your garage, but it can be done under governmental facilities, under situations where it is not risky. But I think that is necessary in order to create predictive models that can help us avoid these kind of things, or prevent, perhaps through DNA sequencing screens, other people from doing that.
Roger.
This was for Yassif.
Sorry, and just before we start a new thread, any other reactions to the comments?
Okay, this is a new thread.
Valda, was your comment in response?
Mine's kind of, because you've mentioned gated access a lot. So there were just two things I wanted to say in sort of response to this. Was one, just as we're thinking about this, to be sure we remember equity, because these frameworks are great, but they're being done by people with the resources to do them. So that's what makes this work so important, because we really don't want science relying on highly resourced for people developing frameworks that should be applied across the board. And then the other thing is, as we're thinking about gated access, to also think about, if that is a strategy, that it doesn't have to be it's gated or it's not gated. In other areas, there are levels that, everyone can have access to this, and then this group can have access to B, and this group gets access to C. So I just wanted to say those two things.
Unfortunately, we only have a couple of minutes left, so I'll ask the panelists if they have any reactions or comments based on what's recently been said. Yep, Jamie.
Thanks. No, I mean, I agree with a lot of the comments around the table. I think, I want to foot stomp Tom's point about looking to the future. We, we're not designing these solutions for where we are today, but we're anticipating that this is going to be a very successful pursuit, and these tools are going to be really powerful, and we need to get ready for that future, because it's hard to do well, and it's going to take time. It's also hard to again, agree that the equity of access is important in building tools that are smart gated access that sort of enable science, but guard against the worst risks. That's going to take effort and time and money. And so we need to start now. But I do agree that, if we're successful, this will be targeted at a narrow subset of tools. Many of the tools probably won't pose significant risks, we hope. And so this is not meant to be a blanket sort of exercise that suppresses science. It's meant to safeguard the benefits by guarding against the worst downside risks, so that the science can flourish.
Okay, Roger, if you'd like to go ahead.
Okay, again, Yassif. So you're calling for, funders to undertake this assessment, risk versus benefit, before they fund things. And the rules for risk assessment, the supposed best practices for DERC and PEP, are actually very explicit, and they're very complicated. Just for risk alone, are very explicit, and they're very complicated. And if done right, it would be a rather thorough statement of possible risks. I've been pushing GPT 4.5 and deep research pretty hard this day, right now, trying to find a single example of a risk assessment conducted according to that framework. None are publicly available, as our AI friends at least help us. I'm wondering if you've seen one, but the fact that there aren't examples of risk-benefit analyses well done that are out there for people to read and see seems to me maybe rate limiting in what you want your funders to do, because people could comment on them and talk about them and decide whether they agreed or disagreed.
Yeah, so I agree. I think, so I would say in both categories, both for DERC EP3 as well as in silico models, the risk assessment problem is hard and unsolved. And we've made a lot more progress in the first category. But actually part of what we're trying to do is solve the problem that you're pointing to. And by developing a pre-funding review set of guidelines to help funders do a better job than they're doing right now, especially outside of the US government, where there are significant gaps. And in fact, Jassy is helping us develop this. We're working first on the EP3 and DERC piece, because even though that's hard, it's more developed. And then next, we are going to look at the in silico piece because we do think that that's a gap, and we're going to have to figure out how to do it. And we hope the community will help us. But yeah, and we're not going to get the perfect solution on the first try. We're going to get like a baseline starting point, and then we'll iteratively refine over time.
All right, James, I'm going to give you the last minute just because there were also a couple of Slido questions that were asking for your thoughts on challenges with DNA synthesis screening and whether it's really going to be sufficient in the future. But I know you had a prior comment as well.
And this is linked to whether it'll be sufficient in the future, and gets back to Tom's point about, you know, not having to build bad things to prove it to ourselves. I do think it's not necessarily kind of a black and white question. I'm skeptical that non-toxic proxy sequences are going to be perfect estimators of whether a model can predict true pathogenicity or true risk. I would love to be proven wrong, but I suspect there's going to have to be some degree of data production to build models that can predict biological risk in novel sequences. And as a company who is going to eventually be in need of using those models to defend our production, I would like to see some kind of framework for thinking through the idea that in narrow cases, you may have to develop data on these sorts of, you know, what you might otherwise think of as dangerous sequences. I don't think non-toxic proxies are going to be sufficient. There's just too many unique mechanisms. And I'll leave it there.
All right. Well, thank you to all the speakers really for providing such rich real-world examples of how computational approaches and in silico models can be used for these complex biological systems, and also to all the participants for your comments. I think these examples set the stage nicely for our breakout room discussions, which are coming up next. So I'll turn the mic over to our committee member, Simone, who will review the goals, objectives, and logistics for the interactive discussions in the breakout rooms.
Okay, thank you, Jesse. So I think a second. All right, so my video is on, my mic is on. This was a great discussion. So our next session, our third session of the day, is going to be, as Jesse said, in the fun, collaborative form of a breakout session. So my name is Simone Bianco. I am Vice President of Hybrid AI and Physics-Based Modeling at Altos Labs. The focus of the next session is going to be to identify the challenges and needs to really effectively safeguard the benefits and mitigate the risks of disseminating in silico outputs through three overarching research themes. The first is molecules and proteins. The second is systems biology and genetic cellular engineering. And the third is epidemiological modeling and biosecurity. And I think they are going to be in the chat as well for you to review. If we can go back to Alex, last slide. Sure. So we're going to ask the groups in each room to go through a set of questions to really prime the conversation for tomorrow on approaches or strategies that should be used to promote high quality science, scientific progress, and the openness of science, as Alex has indicated, while mitigating the biosecurity risks associated with disseminated this research, the in silico research for biological systems. We first ask the groups to review the relevant outputs for in silico research, the dissemination outlets, and the stakeholders that are associated with each output and outlet. And then we ask the groups to answer a series of questions. So first, what are the criteria that should be used to assess and evaluate the research benefits and biosecurity risks? And we want this discussion to really focus on reviewing the existing policies, guidance, and safeguards for physical research. Starting from that, what additional criteria, if any, should be used to assess the biosecurity risk of in silico research for biological systems for each dissemination outlet? We ask the groups to assess if different dissemination outlets really present different dissemination risks. And finally, we ask the groups to report on what do they see to be the current challenges and gaps in assessing and evaluating the benefits and DERC and PEP potential of disseminating in silico research in biological systems. As Audrey said this morning, the groups will have the availability of this online platform called Padlet, and I think you're going to see in a second what this looks like.
You're going to get a tutorial from our colleagues at the academies on using the platform. And in case you didn't think about bringing a computer, we're going to have sticky notes for you, and then we're going to help transcribing this into Padlet. We anticipate you to be able to use the whole time available, but if you have a little bit of time, we would like you to consider brainstorming of about what are the what you can really think it could be implemented now in the near term or in the long term to really fill these gaps and overcome the current barriers to implementing these voluntary guidelines that are also effective. As was said before, breakout rooms interactions are not recorded. Any information shared in this room is not going to be attributed in the final reports. And by joining the breakout rooms, you agree to abide to the Chatham House rules, and I quote, "Participants are free to use information received, but neither the identity nor the affiliation of the speakers, nor that of any other participants may be revealed," end quote. For in-person attendees here in the room and in the overflow room, we have physical rooms available for the three themes. And online attendees who have opted for active participation will have the chance to participate with mic and camera access. Everyone else who is an observer will still have the opportunity to interact with the discussion in the Padlet boards format. You can see that now on the screen. Just as a final comment, in the next few minutes, you're going to be prompted to select a room. And once you're in the room, if you wish to speak, as you was said before, please raise your hand, and if possible, turn on your camera. For the participants in the room, due to the hybrid format that this meeting is ongoing, we request that you are considerate to online participants. And I think this concludes the introduction, and we can break to go to the room.
Yes, so we have a 15-minute break right now. You can refresh, go to a bathroom, go to your rooms. And for the participant, just 15 minutes. And you are going to be added as co-host if you selected to be a active participant. When you move in the room, you will have the access to the mic and the audio, the camera. Thank you very much. And we'll see you at 2:55. Not yet. Online participants and in-person participants, you're going to be situated while we have the break.
So we need to come back here in 15 minutes to get up.
Yes. No, I don't. Well, I think it
Really? Great to see you. Go around. Collaborators, long time colleague. I guess here we're probably did a different breakout session. Right. So, really? No, really? I like looks like bacon today. Somebody had to bring. Yeah. All the feedback. Yeah. Right. Yeah. Oh. Yeah. Oh. Careful it can be, right? Right.
Thank you so much. Hope that was useful.
Well, I wanted the most people to go in the game. I thought it was raining. And now, oh, that's funny too. Well, it's fine. Yes. Thank you. So many good things coming in. So we can go with six. It's 6 months ago. I wanted to.
Breakout. The inequality is getting me. Rooms of weights. You're welcome. Great flowering of dissemination in the year 2020. Idea.
Thank you. I was like, oh shit.
Well done.
So we are getting ready for the next session, just online. A few minutes for everybody to come back to the room and we will be back.
All right, guys, if everyone could start making their way to their seats, we are going to begin our dissemination very soon. Thank you so much.
Yes, I am. Yeah, you're going to do the, yeah, I've got my
So
conclusion.
This is an activity, instead of doing a report out that, we just had a lot of conversations, it goes 4 different questions. It's very hard to summarize everything. So what we'll do here is for everybody who has computers and everybody online is to drop in the chat. So now everybody should have access to the chat, to drop one thing that really resonated to them, one thing that maybe they heard that they had not considered before. It's one thing that is something that they would like all of us to consider as we get into our second day toward thinking about the strategies to overcome some of the barriers that we have talked about today. So I'm going to give you, actually, we have a good, a generous 6 minutes to think about one thing you would have. We really like everybody to participate, especially online. Thank you. Yes, in the Zoom chat. And someone who's going to be the brave person to put the first comment.
There's no lack of brave people in the room, so yeah.
Okay, one more minute.
Thank you. Okay, so we've got some good questions, good comments coming in there. So thank you everyone for being here for all this time with and really a very concentrated effort through the afternoon. We covered a lot of ground today about the benefits and risks associated with the dissemination of computational research. We discussed the extent to which DIRC and PEP frameworks apply. We thought about gaps and challenges that face the different stakeholders and that could stand in the way of responsible dissemination. And I know we got a lot of really important insights put down in the Padlets and a lot of information. And so as a committee, we're going to now go away and get all those Padlets into one document and try and glean through that and come up with key takeaways, maybe key questions that we will use to inform tomorrow's conversation. So this is designed as an interactive and dynamic workshop where what happened today drives what happens tomorrow. So thank you so much for your input, and we hope you'll be back again tomorrow to see what we managed to get out of all those Padlet comments. Thanks so much. But I'm not, sorry, I've forgotten the most important part. So I have to thank our speakers. I have to thank our speakers and panelists for sharing their expertise. Sorry, I missed out the thank you. And I'd also like to thank my fellow committee members and the National Academy staff for doing all the heavy lifting that made this happen. I think it's one of the best hybrid experiences I've had, even with the few glitches. So really, really great. And thank you for joining us. We hope you can take this information away, come back tomorrow, ready to think about, tomorrow we'll be really into the strategy and the solutions. So come back ready to really think about innovative and integrated solutions to address the challenges that were highlighted today. Also, please continue to populate that Padlet board. So, as we work, we'll be watching the new Padlet comments come in. So we'll be reviewing and building on them, and they'll also get integrated into the final report that comes out of this workshop. So thank you very much.
I just want to add two things. One is that please do come back tomorrow. We'll be in a larger room where we'll be a little bit more comfortable. And so, and then the other thing I do want to add a really special thank you to the sponsors. This has been so far like already super engaging, like the level of detail in the conversation, the openness, the collegiality with which these very difficult issues have been discussed, I think it's truly a testament to the ability of the National Academies to bring people together as a convener. And so I just want to make a special thank you to the National Science Foundation for making this happen and facilitating this conversation.
And with this, the meeting is adjourned.